So basically, you just walk on those roads. So on an average day of field work, we walk like 25 kilometers. Just pick up tiger poop, and we're good to go.
You go to jungles, find some poop. Exciting.
Right. Poop.
Exciting.
Yeah.
Hello and welcome to Friendship with Science, a video series where we talk to scientists from India to figure out what's going on in the world of science, understand science, and hopefully have fun. My name is Biswa. I'm a comedian. The idea of this series comes from my dear friend, Mr. Shashi, who's sitting right next to me. I'll try to pronounce his name right. Shashi Thutupalli. Yeah?
Perfect.
Yeah.
Thutupalli.
Thutupalli?
Yes.
Is that correct?
Thutupalli. Yeah.
Thutupalli.
Yeah, very good.
Shashi Thutupalli. I met him at the Chess Olympiad in August last year in Chennai, and immediately it was very obvious that it is not a normal person. The person wants to spread science in India very passionately. To give some context, everybody in the Chess Olympiad who was volunteering was age of somewhere between 16 to 21. This human being is 95 years old. How old are you? 45?
No. I'm 12.
Sticking out amongst the youth. And all the volunteers treated him with a lot of respect. It is very obvious that something great is going to happen. Shashi, please introduce the podcast, the idea, the vision, the man himself, please.
Well, thank you, Biswa. I mean, absolutely. Biswa and me met at this Olympiad, and it was equally crazy that a comedian shows up at a chess Olympiad. And so, I mean, of course, I was deeply excited by the kinds of things you guys achieved with popularizing chess in India, and I thought that was a very commendable effort. What really blew my mind was when you guys had Gary Kasparov playing chess with you guys, and I was like, who are these guys, and why is Gary Kasparov playing chess with these guys? And then, of course, I mean, we started talking at the Olympiad, and turns out that Biswa is also very interested in science, and it seemed like an interesting thing to do together. I mean, there is a lot of exciting and interesting science going on in the world, of course, but even more so in India. And I think this is an opportunity we want to take to talk about that science that's going on in India, but also, learn about the scientists that are doing the science.
Right.
I think this is a fantastic way. So I think this is going to be like you said, a friendship with science and scientists. And let's see how things go.
Yeah, and before we move forward, everybody's curious. Where are we? Shashi?
Where are we?
We're in a beautiful campus of Azim Premji University. Thank you for giving us this amazing support, and very glad to be doing this. Shashi, we have our first guest today. Please introduce.
Absolutely. I mean, it's I would say more than a privilege and honor for me to have my colleague Uma Ramakrishnan. So she's my colleague at the National Center for Biological Sciences, and Uma is as we will see, a deeply passionate scientist. And the first thing that strikes me about Uma is how excited she is about science. So Uma is, in the formal way of introductions in our scientific world, Uma is a molecular ecologist by training. So Uma did her PhD at the University of California in San Diego, and after that did a postdoc at Stanford University, and came back to India with a deep passion and commitment to do all those exciting things here. And I have this book here with me because Uma is, of course, a fantastic scientist, but also a heavily decorated scientist in terms of the award she's won, deservedly so, for the fantastic science that she does. And I thought I will pick a few which reflect not only Uma's work, but also the kind of science that she does and the personality that she brings into this. Okay, so the most recent one is the so-called Molecular Ecology Prize that she won from the journal Molecular Ecology. So it's a very rare and prestigious honor, I think, to be recognized by a journal, which is basically your peers recognizing you, right? And so that's the highest level of honor a scientist would aspire for. And so that's something that Uma has just recently got. She was also honored with the Homi Bhabha Award for Science Education in 2021. So Uma is also very passionate about science education and communicating science to people and so on. The Kalpana Chawla Young Woman Scientist Award from the government of Karnataka. And another very interesting award from the Field Museum in Chicago, the Parker Gentry Award. So you can just see from the spectrum of these awards the kind of people and communities that look at Uma and recognize her. So Uma, it's for me a great privilege to be talking to you in this sort of a setting. Welcome.
Thank you. Thanks. Great to be here.
Amazing. I realized I should have done an introduction for you like that.
Yes.
Well, the difference, of course, is that I am a volunteer at a chess Olympiad and she is actually winning all the awards.
Oh my God.
So
Yeah, I think your introduction is good. You're a volunteer at Chess Olympiad. No, guys, he's a proper scientist.
Yeah.
I don't know what awards are there. I did not know this is a formal introduction.
no, but this is also to sort of give a feel for how we.
Yeah. Got it. Awesome. Very nice to meet you. Uma, welcome and thanks for being our first guest. We had a little bit of chat before, and I understand you work in the wildlife space and you try to understand wild animals. How did this start? How did you get into this?
So I always loved animals. I mean, they're really fascinating. I think as children, a lot of us really love animals, and you look at them and you wonder, like, what are they thinking? What's going on in their lives? And you want to get a glimpse into it. So I was just curious basically about animals and understanding more about them. And I actually did a project on elephants just after my master's. And I would watch them and I thought, like, there's really no way that we can just watch them and figure out things about their lives. Where are they moving? How many are there? Whom are they mating with? What's happening to their populations? And so basically I started wondering about, like, a cheat way to understand more about their lives, and I stumbled upon this field of molecular ecology.
How old were you when you were doing this?
Maybe 20, 19, 20, something like that.
Okay, so in undergrad, basically.
Yeah, okay. Just after, yeah. So, and basically, I realized that genetics or, DNA sequences can tell us a lot about the lives of animals. So I mean, then it was basically after that, I basically use it as a lens or a tool to understand more about animals. I'm also very, I think all of us realize that the world around us is rapidly changing, right? If you just think about it, 96% of mammal biomass on Earth is humans and livestock. So there's
96%.
Yeah, it's crazy.
Wow.
So there's so little wild biomass left, right? And despite the fact that humans are actually small in a biomass spectrum compared to, a lot of other mammals. And humans have changed the Earth like unrecognizably. And it kind of is, I keep wondering, like, how have we changed it? How are we changing evolution? What are we doing to our planet? And so it's kind of a bringing together of those two kind of curiosities which propelled me in this direction.
Do you remember the first time in life you were fascinated with an animal? Like, did you have a pet? Or
So I always wanted pets, but my parents were, like, oh, they're not so clean and stuff like that. So I wasn't allowed to have a pet, but I would constantly, like, take stray dogs on my bicycle when I was a kid, or, go to zoos, always go to any zoo in any city I ever visited. I would drag my parents there because you can also watch animals behaving, and that's really cool. So I've always liked pets. I've not, until recently, I never had a pet, and then I realized, I would have been a better scientist if I had a pet like when I was a kid. But yeah, so I think watching elephants was something else. They are really social, and you can see so much happening which you can relate to, and they're really very similar, right? We all are similar, so that's
When
really
did you
cool.
first see an elephant?
Probably when I was like, we went on several trips, so maybe when I was in high school or something like that.
So that's the animal, like what like street dogs, of course all of us are fascinated by, but what's the one animal that you're very fascinated by when you were younger?
I think I always loved cats. And I really loved, of course, charismatic animals like snow leopards, that I haven't seen one yet in the wild, and elephants and primates, because they have fascinating behaviors and they live in groups.
So where would one have to go for snow leopards?
You have to go to Ladakh.
Ladakh.
Yeah, but you can
But there
see
are
them now.
some in
Yeah,
Ladakh.
Yeah,
Oh wow.
yeah, and also Himachal, but easier to see them in Ladakh.
Oh, wow.
Spiti.
Oh.
Yeah.
Wow. And did you like watch National Geographic, Discovery, all of this? Was it?
Yeah, of course. I mean, it's like how you see the natural world, right? But I guess as scientists, of course, everybody's fascinated by that beauty aspect. But I think as scientists, we try to go further and deconstruct it and understand what is driving it. What is driving those interactions? Why are there more of this here and less of that there? What is happening to these populations in the future? So those questions also were very fascinating.
Got it. Very interesting. So you always had this passion for wildlife and stuff, and then at some point you decided to make it your full career, right?
Yeah.
Do you remember that exact point when you made this decision?
So I was interested in it from high school, and then I realized very quickly that a lot of this work requires quantitative understanding, because you can't just describe wildlife. If you want to understand processes, you need to understand them quantitatively, numerically, right? And so I basically did my undergraduate with math, because at that time you couldn't do biology and math. So I did physics, chemistry, and math, so that I could maintain that quantitative understanding.
Wow.
And then I did my master's in biotechnology, because that was the only way I could get exposure to molecular methods and the tools with which, like genetic tools with which we will understand these
So
things.
your undergrad didn't have biology?
No.
Physics, chemistry, math?
Yeah.
Wow.
And then you...
Yeah.
Okay.
And then basically I finally got to a PhD in ecology,
Right.
because there was no way at that time. Like now there are places like APU and other places, even the ICERs, where you can actually study these subjects together, but that wasn't the case earlier on.
Got it. Sorry, one question. You said you did biotechnology.
Yeah.
Why you did like that? Because I also did
I know.
biotechnology.
It's a
What is
waste
any
subject.
any problem was there?
Waste
Everything
subject.
okay?
Yeah.
Did you enjoy doing
No,
biotechnology?
I hated it. Somehow survived it is all I can say.
Wow. Any biotech boom has come yet? It's coming from 20 years.
Still waiting.
Okay. You have anything to
I
say about
didn't,
biotechnology?
I didn't know this actually.
Okay.
People have so
What do you-
much love for biotechnology.
Yeah. Beautiful subject. But you did, I think once you've done biotechnology, then wildlife also seems like I can live in a jungle also, not that bad. Okay, great. So now you spend a lot of time in jungles, right? And you study a lot of animals. Which is your favorite animal to study?
So I love tigers, and the reason why I'm attracted to them is because they are. So I like things which are difficult, and they're really rare. So a lot of people say, "Why you study tigers? You should study mice or flies or something." And I'm not, frankly, like I love studying things which are difficult to study as well. Also because they are very how should I say, they're kind of nobody wants you to study them, so it's like a challenge to try and understand more about them. And basically India has been pretty successful in tiger conservation, but that's one lens of looking at it. So I'm really curious to understand more about tigers from a different perspective and bring science, not just about numbers, but various other factors. So we do a lot of work. We've been working on tigers for almost 2 decades now. And a lot of what we do is to develop new methods to look at them in a different way, understand what's happening with their populations, how fragmentation of habitats, humans, how they're modifying habitats is affecting their populations, and so on. And some of these things, I also do it because I would like what I do to have some relevance, and the work we do can hopefully at least maybe go into action, or maybe potentially in the long term influence policy and so on.
Can you, like, dogs have very different personality than cats, right?
Yeah.
And yeah, we know because we interact with dogs and cats. I'm sure tigers have a personality. We are told in stories that they're the king of the jungle, etc., etc. But first of all, tigers have a distinct personality as animals, and what is it if they have it?
So tigers are big cats, like blown up cat. So all their behaviors, like they roll, the way they're just like cats. That's why I said earlier, like now that I have a cat, I'm like, if I had a cat earlier, I would have understood tigers better. Right. But basically, in general, cats have very kind of similar habits. And I think tigers, because they are big, they're kind of like nonchalant. They're like, I own this place. They're not really scared of you or anything like that. Whereas there are other small cats which are more cautious because they have predators, and they're just really goofy looking. Tigers, they've got this like puffed up kind of face compared to, say, a leopard, which is like this beautiful, like a streamlined cat. And so I think it's just really fun to watch them because they watch you, like when you're watching them. And it's like very different to, like, if you have pets, right? Like a human gaze versus an animal gaze is really different. So you're always wondering, what are they thinking about me as I'm watching them?
Got it.
So Uma, we started with a bachelor's in physics, chemistry, math, went through biotechnology, PhD in molecular ecology, and now we are talking about tigers. Where are these scales connected? What is the lens through which you have gone through this path?
Yeah. So basically, like I said, DNA is the lens, or genetics is the lens through which we look at wildlife, right? So basically, so
that's where the molecular part,
the molecular part of ecology comes in. Right. So ecology is about studying interactions between individuals and their environment and so on. It's a very broad definition. Right. But how do you do that? Like, if you want to understand, are tiger populations shrinking or expanding on a 100-year scale, right? So actually, the signature of population declines and expansions is in an organism's DNA. And that's crazy, right? If you think about it, that the story of your past is in your DNA. Now, if I say that, if I ask you, Shashi, do you agree with me if I say you're more similar to another human than a chimpanzee? I'm sure you'll say yes. But what is the reason for that? Right. The reason for that is evolution, that you and other humans share a common past. Right. And that past is distinct from the past of two chimpanzees. Right. And so you have more in common genetically. And so basically all of that is written in your DNA. Right. So it goes back to, Darwin's concepts of evolution and a shared ancestry, but also different ancestry. So different
a lot to cover. A lot to cover. First of all, both of you scientists,
zoom out.
What is DNA? That's all. First of all, we need to understand. We have gone very deep into this.
Okay.
But what is DNA in very simple words?
Okay. So basically DNA is a hereditary material for many organisms, and it's the blueprint of life, right? So every single cell in our body or in the body of any other organism has a whole copy of the genome, and the genome is like a book. And basically it's an instruction manual for everything to work. But it's also, like I said, retains a history of the past.
Okay, so We are made of cells.
Yes.
Cells are made of nucleus and a bunch of other stuff.
Correct.
Okay.
Correct. Yes.
The kind of cells that we are made of. Yes.
Yeah.
Some mitochondria, some.
Yeah. Right.
What is this
The
one?
squiggly line.
What is this one?
The endoplasmic reticulum.
Yes. The endoplasmic and some.
Yeah.
Right? And
But
then.
each nucleus has in it a whole genome, right?
Right.
A copy of this instruction manual, this whole book.
So it's like a small thread.
Yes. Twisted and turned like really tight. Because,
Got
for
it.
example, the human genome would stretch actually from here to the moon if you stretched it out.
Okay.
But it's all like.
So genome is the entire thread.
Yes.
And DNA is?
DNA is deoxyribose nucleic acid, but it's basically
DNA is
a
that
component of that. Like,
Hey, you're
Good
saying this is genome, this is also DNA. And then what is gene? What is
very
this
good.
is this is genome.
Yeah.
Then what is DNA?
so DNA is what makes up the genome.
Okay.
The genome is the entire sequence.
Got it.
And basically, DNA can be of 4 letters. You have 4 letters in DNA, A, C, T, G. So in that sense, it makes it easy. Say compared to chemistry, like if you are studying chemicals, there's like gazillions of chemicals, but with DNA, there's just 4 letters, A, C, T, and G. And so if you read it, you just have this long string of A, C's, T's, and G's. So the tiger genome, for example, is 2,400 million letters long. And so you can read the whole genome, and then you can understand various things about tigers, which individuals, etc., etc.
Got it. 1 second. I still don't know what a DNA is. Okay. Genome has many ATGC.
Yes.
The
These
DNA,
ones.
the DNA, think of this, DNA is the molecule.
Okay.
Okay? It's the substrate, the actual physical molecule, which is comprised of this A's, T's, G's, and C's.
So minimum how many do you need for it to be DNA? One ATGC I
Good
put,
question.
it is DNA?
Great question. Yeah, great question. What is the minimum? What is the minimum genome?
Because you're saying genome is made
Right,
of DNA.
right. So the DNA molecule, in principle, has these bases, right? And so if you're right, even if there are a few of these bases present, you would still call that DNA,
Got it.
right? But you might not necessarily call it a genome, or you might not call that a gene, right? And that's absolutely.
So a gene has to be something which can be made into a protein.
Got it.
Yeah. So so the spellings, the arrangement. So basically, the DNA sequence translates into amino acids, which are the building blocks for these proteins, and the gene is a set of spellings which translate into amino acids, which build a specific 3-dimensional protein.
Got it. So let me again get this correctly. If you are watching this, then at least some science we should get correctly. ATGC, what are these called? These are
the bases.
the bases. So A is
Adenosine,
A for
guanine,
apple. Sorry.
A for apple.
Adenosine.
A for apple, T for tomato.
Yeah. But what is, sorry, I don't know. You can tell me. A is
Adenosine,
T,
thymine,
G,
guanine,
and C,
cytosine.
crazy.
Yes.
Okay. So these are what? These are acids? No, these are not acids. What are these?
They're bases,
These
right?
are the bases.
Yeah.
So they're small molecules.
That's
So basically, the structure of DNA is such that there are two there's a backbone which is basically sugars and stuff, and then the bases kind of attach to each other, and it twirls like a helix.
Got it.
So the whole breakthrough was before you could read DNA. So now we can read DNA, we can read it super fast, but we didn't even know what the structure was. Once we figured out, and that's the whole Watson and Crick and others who were responsible for that, and basically Rosalind Franklin, etc. Once you figured out the structure of DNA, physical structure, you could target it chemically in some way to read it, like to open it up and read it.
This is
so now, Uma, what you're saying is that provides you a lens
Correct.
through which
Yes.
you can count the number of tigers.
Yes.
Or anyway,
Yes.
that's a
Yeah.
that's
hello.
a remarkable
1 second. I still don't know what is DNA. Okay, we're still working on this one. I'm not letting you guys go until I understand what is DNA. Okay, so there is a structure of a double helix.
Yes.
which some guy conceived in his dream or something?
No,
Like-
no, no. That is
was
Kekule
Yeah.
and the benzene structure.
Got it, got it, got it, got it. Okay. And this, we have this kind of structure, like like two springs of a ballpoint pen, like that.
Yeah, something like that. The whole thing about the structure was that some X-ray diffraction patterns were used, and basically it's not totally symmetric.
Got it.
So the fact that it was not symmetric allowed them, like for a long time they thought it was totally symmetric and the angle and all that stuff, and then the X-ray diffraction pattern allowed them to figure it out.
So now on this we have ATGC,
Yes.
and they're arranged like you're saying,
The
many
sequence.
millions of
Yes.
these are arranged.
Yes.
And from these DNAs, proteins are made.
Yes.
Like if ATGC is there, accordingly some amino acids will come.
Yes.
And then
from the DNA itself, but based on the sequence
DNAs.
in the DNA.
the
you can-
proteins will form, and then more complex proteins, and then our entire body is.
That's right.
Yeah.
It's correct?
Correct.
Okay. So essentially, the entire information required to create the whole animal is encoded in the DNA.
Yeah.
If all the proteins work correctly, then the animal can be made. This is correct?
That's not entirely
Sort of.
correct.
Yeah. Go ahead.
No, please.
So, I mean, like, for example, if you were to, like, we do cell culture, for example, right? So we can take, say, an animal's blood and grow certain kinds of cells. We can even make stem cells, pluripotent cells. But we are not yet creating full animals from that. Yeah. So in vitro environments, that is outside, like in the lab type environments, we are not yet creating full animals. We're just on the brink of making organs, sort of organelles. I mean, organoids they're called, but this is not my field. So,
But it's also not entirely clear that just from the information in one molecule,
yes,
you can, because there's also, you can create the entire organism or even a
Yes.
cell. Because along with that molecule, there are proteins, there's everything else. So you might need that
Cellular
whole
environment.
milieu also
Got it.
to make everything.
Got it. Okay. How many genomes are there in a cell? Like DNA and genome, now you understand. DNA is a fragment of the genome. We can say that.
DNA is the substrate, the genome is the information that is on that substrate.
Got it.
Think of it that way.
Got
That
it.
there's a physical manifestation, which is the DNA. The information content, which is the genome, is there.
kind of an abstraction.
It's kind of an abstraction here.
Guys, I'm sorry. I'm going to ask again. Until I'm clear on this. DNA, ATGC, I've understood. Double helix structure is there. ATGC is sitting on that one. I've understood. Now, genome has what? It also has only ATGC only, no?
Yes.
Okay.
So think about it this way, maybe, Biswa, that in a computer, or in a computer you have memory, right, which is physically made of silicon, right? But the movie that contains you performing the show is the information that's sitting on that substrate,
Got it.
right? So we won't call that substrate, the chip, as the movie.
Got it.
So you want to think
Got
of the
it.
genome like that compared to the DNA.
So,
Is that a fair?
wow. Genome is like software. No.
Well, so the genome is.
It's like instructions.
It's
So
like
I guess
instructions, yeah.
it is kind of
It's
software, right? It's kind of algorithm.
Got it. Okay. Great. So this is biochemically encoded in the DNA,
Yes.
basically. How many genomes do you have?
We have one genome. Humans have one. Well, I mean, okay. So basically, if you look at, say, the human genome, one in every 1,000 letters, there's a polymorphism or something different, right? So, of course, we have as many unique genomes as there are humans, right? Every individual's genome is unique. But there's also a lot of stuff which is common, right? So basically, again, the chimpanzee example, like any two humans have more similarities in their genome than, say, I have with the chimpanzee, right? So basically, every human's genome is unique. Within our body, most of the cells have the same genome, but sometimes with age, there may be somatic mutations or some, like reasons for small differences in the genome. But overall, that's the whole point, that the genome is identical across cells, despite it being a kidney cell or a heart cell or a whatever cell.
Got it.
Does that make sense?
Yeah. Understanding, but one more question is coming.
Yeah.
Okay, sorry, this is very basic primary level. I think we have invited very overqualified people to explain this. I think you guys are too overqualified for this. Now, humans have 23 sets of chromosomes, is what I remember. All those chromosomes combined are the genome?
Yes.
Okay.
Yes. And this is something which is actually quite confusing.
Yeah,
the funda between chromosomes and genomes.
That's right.
So basically, chromosomes only happen at certain points in the cell's life when the cell is dividing. Otherwise, the DNA is all packed up and bundled and stuffed into the nucleus. But at times, it separates into these, spatially, whatever, like chromosomes.
Yeah, finally.
So you have many layers.
Got it. Okay, now I have understood it. Okay. Can I explain my own words?
Yes.
Genome is like the entire book.
Mhm.
Chromosomes are like some chapters.
Exactly.
And DNA is like the pages.
Or like the letters.
Like letters. Letters I was coming at ATGC.
Ah, okay.
ATGC,
Yeah.
but maybe how the words are strung
Well,
together.
exactly.
Something.
Got it. Very nice. Then what is genes?
Yeah, so basically genes are those stretches of DNA which code for something or encapsulate information to make a protein.
Okay. So like I would come from a set of genes.
Yeah, so the eye is a very complex
Complex
structure, right? In fact, Darwin and his wife argued about it. Because his wife said, only God could make the eye, right? But there's a lot of interesting science also where people have done experiments that, how can you make something as complex as the eye? And basically, lots of, genes together working one after the other can result in a complex structure like
Exactly. The eye has
Yes.
always been brought up as an example
Yes.
of why there's
Yes.
a designer.
Yes, exactly.
And exactly.
Okay. I think I brought up the wrong example. But
Something like hair
Something
color
like hair
is a
color,
simpler
that's a
example,
good,
right?
okay. Now bringing it back. You're mentioning that you work on some tigers that have a different sort of stripes.
Yes.
So the stripes of a tiger are decided by one gene.
Oh, that's again a complicated question.
Okay.
I don't think we know the answer. So the thing is, when we understand genetics, we don't necessarily understand it from this, and this make this. But we understand it from examples of when things go wrong. So this is different, and this gene is not working in that, or something like that. So it's more deductive versus building up. Like we don't know all the genes involved in making, the pattern of a tiger, but we do know that if this gene is not working, something happens to the pattern of a tiger. So we worked on these really beautiful black tigers, which are found only in Odisha. They're always it's Odisha. Yeah.
Yes.
So they've always been a mystery, and the indigenous people who lived in Simplipal actually always talked about these black tigers, and no one believed them. They just dismissed them as they're just imagining it. But then sometime in the late 90s, a youth was actually attacked by a tiger, and he killed it in self-defense, and that proved for the first time that these tigers exist. Their stripes are broader. So, regular tigers have stripes, and you can imagine pattern is like start black, stop black, start black, stop black. But here you have like black. It's like someone has dropped black paint on them or something like that. It's a disruption of the pattern. The pattern is no longer there, no regularity. It's just irregular. So so basically we got kind of lucky because people had actually studied this type of disruption of pattern in cheetahs and in cats, and they had identified a specific gene called Taqpep, which is involved, like mutations in that gene or spelling mistakes, differences in the ATCG spelling in that particular gene results to this disruption of pattern. So we were lucky. We looked at, a zoo family which had black tigers And we looked at this gene and we found, luckily, a single, again, spelling mistake, which causes the Taqpep.
Taqpep is the name of
Taqpep is the name of the gene. It causes a change in the protein. And basically, amazingly, we found also this mutation or the spelling mistake in wild tigers in Simplipal. So it was like crazy because when we worked in Simplipal, we didn't actually see a tiger. We just picked up biological material. We picked up tiger poop and we were able to analyze that poop and figure out whether that poop came from a normal or a black tiger.
Wow.
Super cool, yeah.
Yeah, that's what happens when you do biotech. Ultimately, you can tell if this poop is from normal tiger or black tiger. It's crazy power. Okay, great. Okay, great. This Taqpep, I'm trying to guess the full form. It's tiger and cat's protein end protein. What is it?
It's called trans-aminopeptidase C.
Okay.
Yeah.
There's this famous scientist called Sidney Brenner who referred to these gene names as 3-letter expletives. Because it's always some 3 letters which don't necessarily.
Got it.
But anyway, I mean, this carries meaning. Of course.
Very interesting. Okay, now putting it all together, we know what a DNA is, we know what a genome is, and gene is just bunch of DNAs, like parts.
It's a bunch of bunch of nucleotides.
Let's say chapters in the book. We can say in the
or maybe it's a sentence. Yeah, sentence. Yeah.
Great. So some sentence from say page 2 and some sentence from page 19 and some from 71 combines to give the tiger its stripes.
Yes.
Right? And one spelling mistake is there on page 19 means the tiger stripes are gone.
Exactly.
Crazy. Okay.
Yes.
Got. Understood.
Yes.
Why do the spelling mistakes happen in Simplipal and not elsewhere?
So that is again a difficult question to answer. But what we can do, and that is where evolution comes in. So there's two possible reasons. One is there's something about Simplipal where not that these mutations evolve there, that doesn't happen. Always mutations are random. Like mutations happen randomly, the spelling mistakes. But having that spelling mistake maybe allows you to survive better. So if that was the case, that's natural selection, right? Like black tigers somehow survive better in Simplipal, have more offspring, and so the frequency of the black spelling mistake increases. On the other hand, another possibility is chance. So basically, suppose Simplipal is, a very small population, and imagine you have, black mutations and regular wild type mutations. When you're pulling the next generation out of this, random bag of gametes, by chance, you pull out the black. And so by chance, the black increases. It's not anything to do with adaptation or it's not advantageous in any way. So our work, basically, other kinds of evidence suggested it's more likely to be the latter. So basically, Simplipal is really far away from any other tiger reserves. There's no history of movement, and we showed that again through genetics, that the tigers in Simplipal are genetically distinct. They are very related to each other, suggesting inbreeding. And because of that, it's probably, that is the reason why this black mutation has increased in frequency there, and you see those tigers there and nowhere else. So it's basically, in some sense, human-driven evolution. So, we think of evolution, you think of coronavirus or something like that, or, antimicrobial resistance. But even tigers are evolving, and because of habitat loss, the tigers in Simplipal are now black. And if this goes on, they'll probably all be black. So you'll have fixation, or, where everyone is a black tiger in that population.
Got it. Very nice.
So, Uma, you said that you're able to glean all of this from poop. How is this working?
Yeah.
This is very
So now you're going to get some gory details of poop. So basically, basically feces passes through the intestine, right?
Yes.
And so when it's passing through the intestine, it's pulling off cells from the intestine, and then it comes out of the body. And basically, actually, the top layer of the feces, I don't know whether you've noticed, but it's sometimes like fresh poop, it's kind of shiny. And the reason it's shiny,
it's literally the innards of the gut.
It's the mucus layer, and that actually has epithelial cells. And any tiger cell, tiger genome, right? Tiger DNA. So basically, by just kind of swabbing the surface of tiger poop, we can get tiger DNA. Of course, the poop also has all kinds of other
I was
stuff.
just going to say
It's
that
like a
because
treasure trove of information.
food that it eats,
Exactly.
so it has the
you can
DNA
look,
of all of that.
exactly. So you can look at the prey. So in fact, basically, if you you have prey DNA as well, what the tiger has eaten, and you have bacterial DNA and all kinds of stuff. So the poop actually tells you a lot, and it's also much more easily available, right? When you walk through, the good thing about tigers is they walk on roads, like on the forest roads. So you don't need to like climb trees or something and look for them or whatever. So basically they walk on roads and they mark their territories with poop and spray, which is basically urine. So basically you just walk on those roads. So on an average day of field work, we walk like 25 kilometers, just, pick up tiger poop and we're good to go.
Thank you, Azim Premji University, for giving us the place to discuss. Want to understand a little more on the science aspect of this. So now to put this so far together in context, you go to jungles, find some poop, exciting.
Right. Poop.
Exciting.
Yeah. Yeah.
Come back to your lab, put some poop under microscope. I don't know.
No. We read, we sequence it, right? So we basically read the ATCs and Gs. And in order to do that, we have to do various, molecular biology stuff. Biotechnology comes in there. So different kinds of protocols, experiments, where we, because the poop, the DNA in poop is not that much. It's low concentration. It's also degraded, like a torn up book. And so we need to amplify it or read it using PCR. So we convert like a droplet of DNA to a swimming pool of DNA, and then we can read it. So basically we read it, and since we've already read the tiger genome, we can map this like unknown thing and like match it to that and see, oh, is this a new tiger, same tiger, how different from that tiger, and so on.
You have a full tiger genome, right?
Yeah.
You have understood that this is what it looks like.
Correct.
So you will find a fragment, and can't put any fragment anywhere. No, it has to be a certain length for you to be sure that it is that fragment and stuff. Right. Like you also mentioned, taking a drop of genetic material and making a pool out of it. What is the technology? How does it work? What are the basis of that?
So basically the technology to make a little bit of DNA into a lot of DNA is called PCR, polymerase chain reaction, where you take a template and you copy it and you supply the letters, like these A's, T's, C's and G's are in your experiment, and there's an enzyme which basically it just...
Okay, again, we have gone off. Okay. So first of all, polymerase chain reaction.
Yes.
So it's a reaction.
Yes.
Which forms a chain.
Yes. It forms a chain of DNA.
Got it.
Yeah.
Polymerase is
the enzyme.
Got it.
Yeah.
So there's an enzyme.
Yeah.
Enzyme. Okay, enzyme that's introducing a chain reaction.
Yeah, it's copying DNA.
Got it.
So basically you have the template, or the original, like suppose there is very little, but there is some genomes.
Yes.
Right, in that material. But you want to make many copies of that genome, right?
Yes.
So what you do is you basically, you heat it up, and then this DNA, which is, it's double stranded, right? We talked about it. It splits up, and then the enzyme goes and attaches, and then it takes these blocks, A's, T's, C's, G's, and then copies it, right? And then you basically heat it up again, it splits again. And so exponentially, like you're copying, and you have many, many copies of a specific sentence.
Okay, so it's like, what do you say? We have a bucket of, let's say, magnets, and you have some iron material or something. Like half it is iron and half it is not, or something like that. You dip, and whatever sticks is where the iron is, and then you take that out and then dip again, and in the end you have a lot of copies of the original. It's a very bad example I've given.
Well, in some sense, I guess what you're saying is you have a magnet and you dip it and you collect something, and that is also another magnet with which you can go in and collect more stuff.
Got it.
So each time you go in and dip, you're producing more stuff which you can dip in and pull out, right? So you exponentially increase that number.
Got it.
Right? And so then that allows you to either count or engineer, like what am I saying?
But it's also basically not, so it's not the whole genome. So you can't right now, I mean, you can, there are methods to copy the whole genome, but they are just kind of random. Like, but usually what we do is, for example, when you want to identify individuals or when you want to look for the mutation in Taqpep, you know exactly where the mutation is. You know what's upstream of the, like up before the mutation, the sentence before and the sentence after. And you design a way to basically read between position X and Y, which includes this mutation. And then you basically read it millions of thousands of times. And then once you have enough amount of DNA, you can read that sentence. But you start that process based on your understanding of what that sentence is.
Exactly.
Right? The genome, the scaffold or the assembly or what you know about the tiger genome, you use that to construct this experiment. Because you say, I think this is the word on either side. And then I'm going to read between word this and word this.
My job is to ask dumb questions. You take, you enhance the, let's say you polymerize the
Yes.
DNA that you have. Then what do you do with it?
Basically, then you read it, right? Oh, so you basically now there are these new approaches called next generation sequencing, where you parallelly read many strands of DNA, many words.
So
That's how
like
you
with
read
a
the whole
microscope.
genome. No. It's basically like each base has a fluorescent label, and so it's basically a laser which reads all these pulses of light. I don't know, maybe Shashi, you can.
Yes, it's an entire technology
Yeah, it's
that's
a
been
huge-
developed to.
yeah.
There are multiple ways, in fact, you know.
Yeah.
But yeah.
Yeah. So one of them, for example, this nanopore technology we were
Exactly.
talking about earlier is like it's a cell membrane and there's a pore. A pore is basically something which allows something to go inside the cell, and the DNA strand goes through that pore, and based on whether it's a A or a T or a C or a G, it gives out a specific, light pulse or whatever.
Or an
And-
electronic signal.
Yeah, a signal, and that signal is recorded, and then you piece together like this signal, that signal, this, like you piece together the whole thing.
Got it. Like, is this what spectrum analysis is? Is this not this?
I
Well,
guess.
I mean, I guess you can. I mean,
Yeah, it's
I don't
kind
know
of
what
a
very-
fluorescent spectrum.
Yeah, it's a you use fluorescence spectroscopy.
Oh, so yeah, I think some memories are coming back. Like you do this, and if it glows,
That's right. Exactly.
then it's yours.
That's right.
Then
That's
it's
right.
correct.
That's right. So yeah, I mean, so you have-
Different glows for different A's, T's,
exactly.
C, and
So
G,
you have
right?
different colors.
So different colors, and then basically based on those pulses you put together.
Wow.
And how strong the pulse is, that determines your confidence, whether it's a A or a T. Maybe it's not very strong, then you're not very sure. And that's why you also read things many times. That's called depth.
Yeah.
So you basically like
You
read
increase the
everything,
confidence. in the-
increase the confidence, and yeah.
Got it. So this sounds like many scientists would be involved in this, right? How many? Like, because somebody has to sequence the entire tiger genome. Right? I think it has been done at some point.
Yeah.
Somebody has to identify everything,
Oh yeah.
and then you are not doing guesswork that this is the mutation. That must have been done either by you or somebody else.
No, science is like a social process, right? Like, I mean, I have a lab, and I'm not doing, anything. So it's basically, we work together in teams. Like when I said we walk 25 kilometers, yeah, it's like teams of 3 people each who walk 25 kilometers each. And we do this over months. And then there's people who work with us in the lab. There are PhD students, they have careers in this, they have to finish like a product, amount of research. That's how they get their PhD. So science is very much a social enterprise where we work together in teams and interdisciplinary teams. Like I don't have expertise to know like the chemistry of DNA sequencing. I believe in it, that this machine helps me do it, but clearly somebody developed that machine, right? And they were chemists and engineers and so on and so forth. So science is very inherently dependent on technology. It is very interdisciplinary, and it's a team activity.
Got
a
it.
lot of friendships in science.
Lots of friendships in science, yeah.
Nice.
Lots of friendships.
Very nice. What is your favorite kind of tiger poop? Is it fresh? Is it stale?
So actually, so tigers eat like once in 4 or 5 days because they eat like a a huge deer, right? They'll kill a sambar or a gore. And basically, initially the poop is really loose. And we used to think that, we can't get DNA from it, but it's pretty good. Like we get DNA from most tiger poop. It's really amazing that actually we can get DNA from poop, right? I think it's pretty cool.
Now, can you, sorry, if you just look at a poop, can you know, like, how many days?
Yeah. Because what happens is you have dung beetles and maggots and all that. So that, like, once the poop is old, the dung beetles will start like rolling it up and rolling it away and all. So you can kind of guess, but it varies. Like, for example, in the Northeast, it's kind of humid and it rains a lot. So it tends to look fresher. And, in Rajasthan, it's hot, so it dries up very quickly. But, it's sort of...
Yeah. Inspiring people to take up science. We continue on this path of exploring. But isn't it amazing that most people would just look at it as poop.
Yeah.
But for you, it's genetic gold, right?
Yeah, for sure. So imagine, like, people didn't even see these black tigers, right? When we were there for like 3 months, we didn't see a single tiger, right? And, sometimes you do see them, but it's so rare. So how do you study something that is so rare? And imagine like marine mammals, like whales and all. Like even whales are studied with poop, So basically,
that's hard to collect poop in the ocean.
Right? Yeah, actually you see it. And there are also...
it's just floating there?
Yes.
Oh, wow.
So I think it's pretty amazing that you can study so many species without catching them. Like getting permissions to catch a tiger or even catching a tiger is quite hazardous. And when you have 3,000 or something of a species left, you don't want to stress it out and catch it. And also it's pretty dangerous.
Just tell me this. You're studying all this poop, and now from the poops you can estimate how many tigers are there.
Yeah.
There's a problem with this method. You're not studying the constipated tigers. The numbers might be...
Yeah, so basically, hopefully, that's why we actually walk routes once in 5 days, once in 6 days, because like I said, each animal eats once in 5 days. So hopefully, we will be getting their samples, and anyway, it's sampling. So you're never going to sample everyone. You always have, so basically we use a mark recapture framework. So you walk routes and you sample individuals and you sample them again, and then you use a probabilistic framework to estimate the number of individuals. So you never have a direct count, right? Because you can never be sure that you've sampled everyone. So in most ecological studies, that's why statistics is really important. Quantitative approaches are important because you can't ever say, like if anyone says, I've counted every single animal, that's obviously wrong. Yeah. You can only estimate them.
But that's an interesting question. Do tigers get constipated?
I have no clue.
But that's an interesting...
Yeah, it is definitely an interesting question. And I guess a good way to study that would be in a zoo to see whether the defecation rates are relatively constant or, they vary depending on what they eat. Or
there was recently like this study about, there's a group in Georgia Tech which a few years ago had published this thing called Universal Law of Urination. So they went to zoos literally, and they looked at how long an animal takes to urinate.
Wow.
Okay. Right? And so it turned out to be 21 seconds or so. So they've done something for defecation also.
Yeah. So one thing is that definitely, like one of the things people thought is like, now there's a lot of dumping of cattle, skinned cattle. And there are a lot of wildlife which prey on this. And people thought that they will not defecate as much because the roughage of the skin and stuff, the hooves and all is what makes the poop kind of come out. But they also do defecate. So that might be an interesting way to also look at, whether there are constipated tigers or lions. Sorry.
Okay, coming back to society. Okay, so far what I've understood is a lot of information is stored in DNAs.
Yes.
The simplest way of collecting and studying tigers is by collecting tiger poop.
Yes.
You get all the DNAs, not only of today, but of the past generations also. And maybe that can help you predict the tiger population, how it's changing, etc., etc., etc. So what are some of the findings that you have found, and you have been studying this for how many years now? Decades?
Yeah, almost 20 years.
Almost 20 years? What are some of the key findings?
So basically, I think some of the work we did in central India, which is basically MP and Maharashtra and so on, investigating connectivity or, whether populations of tigers are connected or not, showed us that they are largely connected. So even though populations are far from each other, individuals are moving between them. And this is possible because of genetics. Like you can't like follow animals or collar all the animals to know how they're moving. But certain types of land uses like high traffic roads and high density human settlements are barriers for movement. And luckily, while we were doing this study, there was a plan to expand NH7, which is a highway between like from Nagpur to Delhi. And it bisects two protected areas, Kanha and Pench. Pench is where Kipling wrote Jungle Book, for example. And, there was, because of our work and other work showing that, actually tigers were moving between Kanha and Pench, there was actually a PIL, a public interest litigation, saying that how can NHAI broaden this highway to 6 lanes? Because that means it's going to become a barrier. And we also showed that if it becomes a barrier, the chances of extinction actually increase in this landscape. So basically, based on all of that evidence, the court actually ordered that an underpass be built for wildlife. So it's basically an elevated road. And today, it's the largest underpass in all of Asia. So it was actually a conservation win. So something which we inferred about how basically genetics was used to infer whether tigers are moving or not, and what types of changes or development might impede tiger movement or stop it. And that was then used kind of proactively to suggest mitigation.
Got it. Lot of ground to cover. Okay. No, it's great. My question to both of you, what I'm understanding from this is that diversity is good for the survival of the species, right? If the gene pool is varied, then it survives more. But if you isolate them into small territories, then the likelihood of them getting wiped out increases.
Yes.
Why? In simple terms, why?
So basically, it's not just because of genetics, it boils down to stochasticity. So when you have a very small population, environmental effects could wipe it out, or demographic effects, a bad year where one individual reproduces more, the other doesn't, and so on. Or genetics. So you just happen to have a poor combination of genetic variants there, and they're just not enough for future challenges which may come up. So basically, the minute you have low variation or small numbers, you lose the ability to survive into the future. And basically, empirical studies, studies on many species, modeling studies, as well as experimental studies, all indicate higher probabilities of extinction of small and isolated populations. And as we have habitat fragmentation, as I said, humans have impacted so much of the earth. If you think about it, 4% of India is protected land area wise, right? Globally, the guidelines are 10% for any country, but India has much smaller protected areas. So all of our wildlife is really sequestered into these islands between seas of people and seas of human kind of development.
Got it.
Yeah, it's basically a small number game, right? So that's one possibility, of course, right? So if there were 100 tigers and suddenly you have isolated 3 of them, the chance that some calamity comes and wipes out those 3 is very high. And if they were genetically distinct from the other, then you've just completely lost that genetic diversity in one shot, right? So, even when we think of population, we think of large numbers, but literally we are talking about a few thousand tigers here. So that's just, it's like...
How many tigers are there in India?
So, there's a lot of debate about that, but 3,000 to 3,500, something like that.
And some 2-3 constipated prankster tigers. What no? Yeah.
My DNA ain't going to NCPS.
Under the radar.
Yeah. No other card tigers. I don't poop. or they like dig up and then I've learned from dogs. Anyways, 3,500 tigers are there, you're saying. How are they distributed across India?
Yeah, so basically there are 53 what are called tiger reserves. A lot of tigers in MP and Maharashtra. A lot of tigers in the Western Ghats, Karnataka, Tamil Nadu. There's a bunch of tigers in the Tarai, which is basically UP, Uttarakhand. Some tigers in Rajasthan, Ranthambore and the population we've studied, and basically not many elsewhere. So very few in the northeast, a few in Odisha and so on. So areas where the numbers of tigers can increase are like Jharkhand, Chhattisgarh, Odisha, West Bengal. But in other places like in MP and Maharashtra, there are lots of tigers and they're at carrying capacity. That means there isn't really much space for more tigers.
So there are some in the north. You're saying the western part of India has Western Ghats, sustained population it has. We don't need to do anything. They'll keep sustaining basically. But on the eastern side, there are problems.
Yeah, so basically there's something called the empty forest syndrome. So where you have forests, but you don't have mammals, you don't have large mammals, you don't have carnivores and so on. And this could be because of hunting, absence of livelihoods, and things like that. So basically, ultimately, conservation also comes back to people. I think we need to support our people so that they can also have other livelihoods, other options for what to do, which don't depend on bushmeat or hunting and so on and so forth, and protect. So basically, tigers are like any carnivore. If there is prey, they will come back. And so if we could somehow, even when you're talking about Simplipal, it's in Odisha, of course, a quick fix would be to introduce individuals, airlift them or something. But on the other hand, if we could, plan for connectivity in the future, allow for corridors for tigers to say disperse from Chhattisgarh, say, into Odisha or something like that, that would be the ideal scenario going forward.
Are they actually moving that?
They do
that
move
much?
long distances, right?
So we are talking about
hundreds of kilometers. Hundreds of kilometers. So basically, as of now, Simplipal is about 500 kilometers from any source population which has like excess tigers, like where there's enough individuals that they move out. So I think it's something which it's hard because on one hand development is crucial for people, right? And places like Chhattisgarh and Odisha, there is a strong need for development. So how do we balance these things? It's a very complicated and a difficult question. But biodiversity is critical for our well-being. I mean, the air we breathe, the water we drink, like there's no technology which can create drinking water. Israel is doing it at great cost, but it's not easy at all. And the fruit we eat, everything is very much linked to biodiversity.
I will like to irritate you here.
Okay.
One irritating question I'll ask. Because in science we ask every question.
Of
right?
course.
As in you guys, I'm not science. What happens if all the 3,500 tigers die? How does it change my life? A tiger living in jungle, not doing anything for me, dies. What happens?
So basically, tigers exert top-down control of ecosystems. Tigers or top carnivores like tigers. So basically, the number of tigers keeps the number of deer or herbivores in control, and keeping the number of herbivores in control affects the vegetation and the rodents and so on. So, for example, there are really nice studies in Africa which show that when you lose the large, if you don't have many herbivores, you have lots of rodents, and rodents, for example, carry disease, and you can have like zoonotic, spillover or something like that. So basically, this ecosystem, the balance in the ecosystem and the food chain is maintained top down as well as bottom up. And tigers are basically species which kind of control the food web and the whole ecosystem. Other than that, tigers are basically, most of the world's tigers live in India. So if tigers are to survive, they will survive in India. And so in that sense, India owes the world, tiger conservation is something which it's India's responsibility globally as well.
Sorry, one question.
Yeah.
If tiger's only job is to kill a certain number of deer,
Yes.
can we not write an AI that kills that deer? Do we really need Sorry, I have to irritate. I have to get to the bottom of this. Can I write a very, would take some Boston Robotics tigers,
yeah.
program them to kill exactly in that pattern? Then will the ecosystem be okay? Or tiger doesn't need more?
So, I mean, I guess it's not just about killing deer, but I think that, tigers, they.
I mean, wasn't there this great example in Serengeti, where they had essentially vaccinated cattle nearby because the cattle were carrying some virus, and that vaccine spilled over into wildebeest, and the wildebeest survived. And because the wildebeest survived, the grasslands were, eaten up. And because the grasslands were eaten up, there was no grass to catch fire. And because there was nothing to catch fire,
the forest
came back.
Yeah.
So, and you see, I mean, we're talking now about a virus that was controlled, right? So, as I guess, Uma, you were saying that this poop itself has a lot of other stuff, means there's a whole ecosystem within the tiger itself. I mean, of course, in the Serengeti, there is this linkage all the way, from the scale of a virus to the entire ecosystem. And Uma, you were saying that, this poop is full of
nutrients, right? So there's also nutrient cycling.
Exactly, yeah.
And this conversion of grassland to forest is something which is a very important thing to also consider today. It's what's happening also with global change, and also, in India especially, we are very focused on forests. And we actually ignore grasslands. And that's why, for example, the great Indian bustard, probably it's going to be the next species which we lose from India, because we've just built all over the grasslands and destroyed all the grasslands because they're considered wastelands. They're not considered important habitats.
So basically, is it possible to even predict what happens when one species disappears?
Yeah, it's very difficult. So there are these theories of cascade effects that, basically, so imagine like you have a food web, right? So one way to look at it is, A is connected to B and so on. Imagine you can do perturbation analysis where you can cut off one, linkage in that. And you can actually study mathematically what happens to this food web when, one of those things is removed, right? So that's how people have done it theoretically. But unfortunately, some of these things are very hard to do in the real world, right? You can only study it when you have, say, extinction of something or something else happens. Like when you opportunistically, we can't do experiments when we work on nature sometimes as easily, especially these kind of experiments. But there's an extinction happens or something happens, then we can understand that situation and we can make hypotheses or predictions about what might happen more generally. Does that make sense?
Yeah. Now, there's a story, I'm sure you guys are aware, but just to repeat, when plague was spreading in the 16th century in Europe, they thought the cats are spreading it, killed all the cats. So the rats went mad and plague spread even more, right? Essentially what I'm asking is it possible that there will be a big disaster that affects the entirety of human population just because we lose 500 more tigers? Is it possible?
It's possible. I mean, I can't say that it's definitely going to happen or whatever, but it is possible because, like I said, if there is top-down control of ecosystems and if we lose tigers, that has many downstream consequences. Also, once you lose, there's also socio-political implications. Like because tigers do generate revenue from tourism. If you, for example, lose, tigers and you lose the Sundarbans, all of your, water purification of the city of Calcutta is gone, right? So the thing about environment is there's a lot of what are called negative externalities. You don't appreciate what you get in terms of, free services from the environment, clean air, water, and so on. But when it's gone, when nature is gone, you're hit by a catastrophe. And that's when it's like costs you a lot, but what you're getting from it is free, if you know what I mean.
Yeah.
Maybe you've heard of this very common thing that the flutter of a butterfly in South America can cause a tornado somewhere in the other corner of the planet. So,
Linked systems.
These are all very highly linked in a highly non-linear way.
Yeah.
Very hard to predict what can happen. Right? So it's not easy to say what exactly will happen if 500 tigers are gone, but presumably something bad will happen.
Yeah. Got it. So let's say you go to Simplipal or you go to Ranthambore and you find bunch of tiger poop and come back, you study that. Now you know the past of that ecosystem, what is happening in the recent past also you come to know. Then you predict that, oh, maybe there's a problem.
Yes.
If you don't stop it,
Yes.
something will happen.
Yes.
Right?
Yeah.
And then you basically call up somebody in the government and say, don't build that highway.
No, I'm not so powerful. But hopefully, like the thing is that I think that decisions in society should be made on evidence, and we provide that evidence. Like, I'm not probably the person who's going to call up someone, but as scientists, our role is that the data should be there. The evidence should be there based on which you can make a decision. So if you decide to build the highway and increase your probability of extinction by 50%, that's on you. We have shown you that that's what's going to happen.
Got it. Yeah. So you call, or somebody calls. Somebody calls somebody and says,
hopefully calls.
Somebody
It's not even obvious that somebody calls.
Just somebody hopefully calls somebody and says, don't build that highway. And then they say, why? And we say, because some tiger poop has been collected. And it doesn't look very good. Yeah. And hopefully that highway doesn't get built and the tiger movements remain.
Yeah, or the highway is built in a different way.
Yeah, it's
So I don't think we should say like no development at all. That's not an option, right? And that's not fair for me and you sitting in, cities. So I think that what would be nice is to look at alternative scenarios or ways to basically minimize, biodiversity loss or,
incorporate that into design.
Incorporate that into design. Yeah.
Also very interesting that it's not enough just to have isolated patches of conservation.
No. So basically, these ecosystem functions, right? Regulation of cycles and, how ecosystems work. They need large landscapes. They cannot function at small scales. You can have a microcosm of an ecosystem in a test tube, but that's not functional like the Serengeti, right? The Serengeti has immense potential for services and to function as an intact ecosystem. And the problem in India is like, if you take all of India's protected areas, they're just 3 times of Kruger National Park, which is one protected area in Africa, right? So we have really tiny patches which we are working with. But despite that, we have amazing biodiversity and amazing, still intact habitats. And that's primarily because, of the people. I think people in India really value biodiversity and wildlife. And that's something we shouldn't lose. We shouldn't make it an us versus them. And, we shouldn't lose that goodwill that we have.
What do you foresee happening in the wildlife world? What are some problems we face right now? How are we going to overcome? What are your future projects? Something like that.
Yeah. So, from a practical perspective, applied conservation perspective, we have a lot of conflict now, a lot of cattle lifting by tigers and so on, and also human attacks. And as the number...
Humans are attacking tiger is attacking. Sorry,
Yeah. Humans are getting attacked by tigers.
Big problem. I'm sorry.
Yeah.
Humans also have been
Humans have also attacked tigers because their pelts are valuable and so on. But so basically one issue is to try and manage those situations. And I think there again, for example, if we could take some of these techniques and tools like sequencing into the field, not like bring it to the lab, take it into the field, and be very dynamic. In general, science is kind of slow, like we just potter around and do things over years, and that's not the time scale at which you need to solve problems on the ground. So that would be really nice. But I'm personally, I'm going to work. I've always been fascinated by the past, so I'm going to do some time travel, and I want to understand. In the past, even 100 years ago, tigers were all across India. Like we said, now they're in a few places, but they were all across India. And I'm really curious to know what we've lost. And the only way to find that out is to actually go back in time and look at, the genetic variation in tigers from the past. And so we're going to work with trophies. There was a lot of hunting, both in the British Raj times, but also post-independence. And a lot of those, skins and skulls are in people's living rooms of the erstwhile Maharajas, in museums. And I'm hoping to, get a little bit of skin or some bones from inside the nose, and again, get DNA, read it, and compare it to current tigers to understand whether what has changed, but also have these bad mutations which are potentially harmful increased, and what is going to be the trajectory of tigers in the future.
So the DNA is going to remain intact in these things?
Yeah. So, I'm sure you remember, Shashi, last year's Nobel Prize went to this guy called Svante
Exactly.
Pavo.
Yeah, of course. Yeah.
And he basically sequenced archaic human genomes like Neanderthals and,
Exactly.
others, Denisovans who were our close, relatives in a way. And that's what he got the Nobel Prize for. And so, now we can really not only read DNA from, extant individuals, but also from the past. And that's just an amazing idea, right? To go back in time. So yeah, I'm pretty excited about that.
How are you going to find these people who have the tiger remains?
So it's really funny. So basically, there was a lot of people shooting tigers, and they had to all go to a taxidermist. So basically, it's a dead animal, warm and all that. So what they would do is they would ship these dead bodies to most of the taxidermy or the preparation of this flat skins and stuff was done by Vanningen and Vanningen. It was a company and two brothers in Mysore, and they basically had 14 ledgers where they entered all this information of the skins that they have prepared. And there's just this one guy called Pat Morris from the UK who came and then they died, and then their whole estate was in a big legal battle and stuff. So they are no more. But these ledgers are with, copies of these ledgers are with this guy called Pat Morris, who's in the UK. So I'm hoping that he'll be willing to share this so we can actually trace back, for example, tigers around Simplipal, or, it would be really nice.
Tiger trophies.
Yeah, trophies. Yeah. And who has them, and can we actually write to them? So it's going to be a bit of a wild goose chase. But also there are a lot of erstwhile Maharajas' palaces, where these trophies are there. So we'll have to see. We've never done it. They may not work, or, who knows, but we'll have to try.
Wow.
So we can bring the past back.
Hey, I saw some 4 skulls in your house. Sorry, it's a human skulls. That's okay. You're not interested in that. No? Tiger skulls.
She's threatening to bring the past back. So
yeah, it's not just me, right? I think there's a lot of work on this concept of de-extinction. So basically people feel that, humans have been responsible for extinction. So we need to correct the wrongs or the misdeeds that we have done. There's many, but anyway. So the idea is, for example, to bring back woolly mammoths. And there's even discussion, for example, of, why not bring back white tigers? Because they once roamed freely in India. So these are also ethical debates, just we've recently brought the cheetah back. So there's a lot of debate about whether we should have done it or not and all that stuff.
It's not just ethical debates, right? Because it's also an ecosystem perturbation experiment in some ways.
Correct. But I personally think that the future is going to be a lot about these kind of experiments, because we don't have anything natural anymore. Everything is modified, everything is tampered with to different extents, right? And so it depends on what we want. Do we want the cheetah back or not? Do we want mammoths or not? Who wants it? And basically then
the future
many
is bringing the past back.
Correct. Well, it's about I think the future is engineered.
Which is
So in that sense, your example of the
Boston
AI
robots.
Boston robots which killed off deer, I think that it might be that we see things like that in the future. And maybe I'm being like a bit crazy, but I think that this clinging to this view of this natural, wonderful, acha tha, is no longer going to happen. Technology is here, the changes are here. Humans have changed the earth in unrecognizable ways. What we have to see is what is the future going to be and how do we want it to be.
So all this from poop and then for the future of the planet. Yeah. Yeah. Isn't science is insane sometimes, all the times actually. Thank you so much for being part of our first episode. Thank you, Shashi.
Thank you, Biswa.
Thank you. Thanks. It was
Thank
really
you.
fun. Yeah.
Yeah. Okay. We will be doing many episodes of this. What we want from you, the audience, is if you have any particular topic that you're very interested in, or any Indian scientist you want us to get on board, and any questions you want to ask scientists, please comment. Please comment everything. We're not promising we'll solve it in the next episode, but all your questions, all your scientists, everything we want to answer, and we'll eventually do it. So thank you for watching our first episode. We'll be releasing many more episodes of Friendship with Science. Some love for our expert and for our guest in the comment section. Thank you. See you next time. Like, share, subscribe, and bell the ring icon. I don't know what is now YouTube. Ring the bell icon. Whatever. Do whatever other YouTubers have told you to do. Do all that. Enjoy.